Dr. Eric Kaldjian is Chief Medical Officer at RareCyte. He trained in anatomic pathology at the University of Michigan and subsequently at the National Cancer Institute. His pharmaceutical experience at Hoffman La Roche and Parke-Davis Pfizer encompassed discovery research through full clinical development positions with a focus on translational medicine. He has directed clinical genomics programs at GeneLogic, was Chief Scientific Officer at Transgenomic, and was Medical Director for Companion Diagnostics at Ventana Medical Systems before joining RareCyte, where he oversees scientific and medical applications of its rare cell detection technology. He is an associate member of the Biointerfaces Institute at the University of Michigan, and it is a pleasure to welcome you to the podium, Dr. Kaldjian.
Thank you very much. I'd like to thank the organizers for the opportunity to be able to discuss with you today the promise of liquid biopsy and to present efforts to deliver on that promise using the RareCyte platform. In this program, I will be presenting materials for educational purposes as described here in this slide. Also, it's important to state that all products being presented here are for research use only at this time and not for use in diagnostic procedures. The learning objectives for today are to understand how rare cell analysis, including liquid biopsy, can be incorporated into diagnostic pathology to extract clinically important information, as well as to understand the value of phenotypic and molecular characterization of individual rare cells within liquid biopsies and other specimen types. The approach to accessing tumors for analysis has evolved from highly invasive to increasingly less invasive. And yet, at the same time that biopsy sample volume has decreased, the diagnostic information required from the sample has increased. With liquid biopsy, we have a non-invasive sampling method with very few cells. The challenge then is to extract information from these few cells. First, a little bit of the past of liquid biopsy. The term liquid biopsy has quite rapidly entered the vocabulary of diagnostics. Its original meaning was to describe a way to access the cells of a tumor indirectly by capturing rare tumor cells that circulate in the blood and thus allow a real-time investigation of the cancer via blood sampling. Before discussing the future, let's actually go even further back to its original origins. As many of you know, the origins of the field can be traced to nearly a century and a half ago in the famous report from Thomas Ashworth in the Australian Medical Journal. In this paper, he described a patient that had many tumors visible in the skin throughout the body. He investigated the blood to try to understand how so many tumors appeared. The tumors had cells with an unusual appearance, and Ashworth saw similar cells in the blood, allowing him to infer a relationship between the tumor cells and the cells he found in the blood. Here are the figures that he drew of the tissue and of the blood cells. Below is a micrograph of a chordoma, which is likely what the tumor that Ashworth described as a tumor of chorda dorsalis in his paper actually was. You can see a similarity between the cells in the micrograph and in the drawings. Microscopic visualization of the cells is what actually allowed Ashworth to make his investigation and arrive at his seminal conclusion. That was the past. The present has largely been influenced by the cell search system. This can be considered the first generation of liquid biopsy. Cell search accomplished remarkable milestones for liquid biopsy. Notably, the prognostic significance of CTC counts in epithelial cancers. The future of liquid biopsy will involve expanding the breadth of diagnostic analyses to match what can be obtained with tissue biopsy. CellSearch has robustly addressed the first level of tissue biopsy evaluation, namely identification and counting of circulating tumor cells. A second level of evaluation is phenotypic characterization of malignant cells, as well as potentially immune cells in the tumor microenvironment. A third level is the molecular analysis of individual cells. Here's an example of what next-generation liquid biopsy will look like. These are unusual cells that were found in the blood of a young woman. You can see in green that the cells are expressing cytokeratin. This woman had no history of cancer. She did, however, have a history of pregnancy. These are, in fact, fetal cells circulating in maternal blood. We know this because we isolated them individually and demonstrated on a single-cell basis that they were male cells in a female host. After removal of individual cells, whole genome amplification, and CGH array analysis, we can see in the left panel the presence of a Y chromosome in the cell in a maternal sample. Similar analysis of blood from a woman known to be carrying a fetus with trisomy 18 confirmed the presence of the additional copy of the chromosome by both CGH array and next-generation sequencing, as you see in the panel on the right. Even more remarkably, a 2.7 megabase microdeletion was confirmed in a circulating fetal cell in another individual. This demonstrates the feasibility of using fetal cell analysis to eventually replace amniocentesis or chorionic villus sampling for testing for genetic abnormalities. In effect, this is a liquid biopsy of the placenta, since these cells have trophoblast origin. As we know, traditional tissue biopsy collects tumor samples by an invasive procedure, processes the tissue, typically using formalin fixation and paraffin embedding, to a slide. Sections are then stained to identify the cancer and further characterize it by IHC phenotyping, and biomolecules from the tissue sections may be isolated to perform molecular analyses. At RareCyte, we have developed a platform that is in many ways a direct descendant of Ashworth's analysis 150 years ago. It is modeled after established diagnostic pathology workflow to put cells on a slide. Once on a slide, what can be done with the tissue section can now be done theoretically with rare cells. Cells may be identified, characterized by phenotype, and then analyzed at a molecular level. The first step in the process is called AccuCyte. It's sample preparation. AccuCyte separates and collects the nucleated fraction of the blood that contains the rare cells of interest within the Buffy coat. It's a user-friendly system that exploits the characteristic density of nucleated cells, which are transferred then to a microscope slide. Since the Buffy coat is collected in the process without washes, the plasma can also be collected from the same blood sample. Once on the slide, the cells can be stored on that slide in the freezer for a long period of time. The prepared slides can then be placed into automated immunostaining instruments for immunofluorescence staining with multiple markers. The standard markers that we use to identify CTCs include epithelial cell markers, cytokeratin and EpCAM, a leukocyte exclusion marker, CD45, and then a nuclear stain. The use of automated stainers eliminates staining variability and increases control of conditions to allow for amplification of low expression targets, and as we'll discuss more later, up to 6 markers to be simultaneously assessed. Stain slides are then imaged with RareCyte's SiteFinder automated fluorescence microscope. SiteFinder is a precision-engineered instrument that scans a complete slide in about 11 minutes and has up to 6-channel imaging capability. The scanned image is analyzed by an image analysis algorithm in about 10 minutes. The algorithm identifies candidate target cells that have the characteristics of the cells of interest and presents them to a reviewer for confirmation. The algorithm ranks candidate cells based on the defined parameters that determine how likely a cell is to be a true CTC and gives it a score then between 0 and 100. The highest scoring cells are presented to the reviewer first. This increases consistency of reviews, decreases user or observer fatigue, and also speeds the whole process and makes it more efficient. Integrated within the SiteFinder instrument is the site picker, a built-in module that mechanically dislodges individual cells and pulls them into the needle tip of the instrument. Cells can then be expelled into an imaging tube, allowing visual confirmation of the individually retrieved cells before single-cell molecular analysis. The site picker is highly automated and is designed so it does not require a high degree of technical skill. Here are some examples of lung and breast cancer circulating tumor cells imaged using the 40X objective, also part of the SiteFinder instrument. You can see that there are many cytological details visible in the cells. Here's a case study that used the RareCyte platform for the monitoring of CTCs, as well as single-cell isolation for genomic analysis. In an innovative clinical study directed by Tony Blau at the University of Washington, nearly 50 CTC analyses over 9 months were performed on blood from a patient with triple-negative breast cancer. Her CTC count paralleled her clinical course. Her baseline of more than 1,000 CTCs per ml dropped 100-fold during platinum therapy, and then climbed again as she progressed. She did not respond to single-agent targeted therapy with crizotinib, and that was chosen because the patient had a ROS1 mutation that was confirmed in circulating tumor cells that was theoretically a potential driver of the cancer. She had a brief response to eribulin at the end of her course that was also reflected in a reduction of her CTC count once more. Surprisingly, when she died, she had a lower CTC count than she had had 2 months previously. This may be because before she died, she had very large clusters of circulating tumor cells in her blood. This is one of the most dramatic and notable clusters that we found. You can see that there is tremendous heterogeneity of size and shape, as well as heterogeneity of EpCAM phenotype. EpCAM is marked in red. You can see some cells are no larger than a lymphocyte and are cytokeratin positive but EpCAM negative. Others are very large with very prominent EpCAM staining. The cause of this patient's death was respiratory failure. Here's a microscopic image of her lung at autopsy. Although the alveolar air sacs are open, the arterioles are plugged with tumor cells, emboli that kept blood from circulating through the lung. This was a case in which a patient died quite literally from circulating tumor cells present in her blood. One of the advantages of the archivability of the samples is that we could go back to samples that were collected earlier and perform molecular analyses. We retrospectively isolated CTCs for single-cell genomic analysis from 6 time points during the patient's course. Individual CTCs were picked with site picker, single genomes amplified, and whole exome sequencing performed with our colleagues Anu Madan and Kelly Howard at Covance Genomics. Multiple bioinformatic filters were applied to generate a list of sequence variants for each cell. The results are shown here in this figure. Each column represents a single CTC grouped by time point. Each black bar represents a mutation that was identified in that CTC. The mutations can be classified into 3 groups. First, a static group that remains unchanged during the course of therapy. You can see that group at the top of the figure across all time points. Sample-specific groups that are unique to each time point. So for instance, you can see at day 6, at the bottom of the group of mutations, there are variants present that are not present at day 91. And then in addition, there are intermediate groups that have variants that are short-lived but are present across multiple time points. Note that the cells on day 91 are not directly derived from cells on day 6, since they do not include many of the mutations that are found on day 6 cells. This suggests that they are different branches from the same clonal tree, related at the trunk level, but not directly springing from the previous set of CTCs that were evaluated. We further investigated the variants to determine how many were predicted to be deleterious or were known reported cancer drivers. Cancer driver mutations increased after exposure to therapies that produced clinical response. You can see that at day 91 relative to day 6. Over time, you could see that as the patient began to progress, the number of cancer drivers decreased, and then after the successful brief treatment with eribulin, with CTC count decreasing, the cancer drivers, again at day 258, came up. Interpretations of these data are really quite intriguing. One possibility is that the cells with increased cancer driver mutations represent malignant cells that are resistant to the current therapy. RareCyte has built a framework for custom assay development that utilizes the multi-channel capability of the platform. We call this the 4+ framework. 4 fluorescent channels are used for the identification of canonical CTCs using stains for nucleus, the epithelial markers, cytokeratin and EpCAM, and the white blood cell exclusion marker CD45. This leaves 2 additional channels available for biomarkers of specific interest. High amplification is possible for targets that may be expressed at low protein levels. In fact, if cytokeratin and EpCAM are combined into 1 epithelial channel, then 3 additional biomarker channels are available for investigation, further extending the depth of characterization available for rare cells. The following slides demonstrate how the 4+ framework allows biomarker investigation in prostate cancer. Here's a 5-channel assay that adds androgen receptor, or AR, to the 4 channels. The androgen receptor is a principal driver of prostate cancer. Here you can see that its staining location is predominantly nuclear in this cell line spiked into blood. The antigen splice variant ARV7 has been associated with resistance to endocrine therapies such as enzalutamide and abiraterone. Here you can see its presence also in a nuclear location. This image is of a 6-channel assay in which prostate-specific antigen and antigen receptor have been added into channels 5 and 6. The other markers are not shown in this particular image. Here's a 6-parameter assay applied to a sample in breast cancer. In this assay, the immune checkpoint regulator PD-L1 is identified in the 5th channel, and the interferon regulatory factor 1 and interferon gamma-induced transcription factor that regulates expression of PD-L1 is in channel 6. Recent studies comparing PD-L1 and IRF1 tissue staining have shown that IRF1 had a higher predictive value than PD-L1 in determining response to checkpoint inhibitors against PD-1 or PD-L1. You can see here that the localization of IRF1 is in the nucleus of cells in a CTC cluster. The promise of liquid biopsy really is to make possible via blood sampling what was previously available only via tissue biopsy, and this includes companion diagnostics. Here's an example of a drug target marker that RareCyte has developed with a pharma partner. Here you can see distinctly in green the presence of the drug target on a cluster of cells that were collected from a patient-derived xenograft model and spiked into human blood. The same assay was subsequently applied to blood from cancer patients. Here we see a cluster of cells that express the target of the drug under study. This assay is now being implemented in clinical trials of the drug. The technology that RareCyte has developed for identifying circulating tumor cells in liquid biopsies may also be rationally applied to other sample types in which rare cells are present. Often, a definitive diagnosis is not possible in small volume cytology samples such as fine needle aspirates, and additional biopsies are often required. Here's an example of a fine needle aspirate sample of the lymph node in a patient with classical Hodgkin lymphoma that we evaluated in collaboration with Jonathan Fromm and David Wu at the University of Washington. Using a 6-parameter panel, cells with a phenotype that included CD15, CD40, and CD30 were identified using SiteFinder and then picked individually using SitePicker. Analysis of the heavy chain of the B-cell receptor identified PCR products with precisely the same length from these two cells, providing very strong molecular evidence that the cells were in fact from the same clone, thus confirming that the cells identified were in fact likely to be Hodgkin cells. The RareCyte platform can also image tissues by immunofluorescence, and individual regions of cells can also be picked by SitePicker. Methods for molecular analysis of cells picked from tissue are under development. This will allow the correlation between phenotype, including morphology, of cancer cells and their genotype. Furthermore, molecular analysis of atypical cells could be performed to determine whether or not they are, in fact, malignant. Finally, the platform was technology that was designed to identify cells from solid tumors in a liquid biopsy may also be used to identify minimal residual disease in liquid tumors. Here's an example of CD138 positive malignant cells identified in the blood of a man with multiple myeloma. I envision that in the future, samples that are initially non-diagnostic due to insufficient numbers of atypical cells could undergo rare cell picking and molecular analysis. In many of them, molecular alterations that indicate presence of cancer could effectively turn them into diagnostic samples. It has been a pleasure to speak to you about the promise of liquid biopsy and how methods developed for it can be applied to other sample types. Thank you very much for your attention, and I would be pleased to answer questions.
Well, thank you very much, Dr. Kaldjian. Great presentation. We do have some questions, and we'll go right to them. Is liquid biopsy useful for mesenchymal tumor or only for carcinomas?
That's a very good question, and let me start in give a little bit of background first. And our technology allows the identification of any rare cell that's present in the blood, as long as there are markers for that rare cell that can differentiate it from the other cells that are present. So if one were to, for instance, look for circulating melanoma cells, one would use a panel of markers that included melanoma-specific markers. And that's quite possible, and we have developed such prototype assays. In addition, if one were to look for, say, circulating sarcoma cells, again, the challenge there would be to come up with a list of antibodies that would differentiate your sarcoma from other cells that were circulating in the blood. I will just add that with the great interest in epithelial cancers that undergo the epithelial to mesenchymal transition, we have developed a combined epithelial mesenchymal assay which includes Vimentin in order to be able to identify such cells.
Excellent, thank you. Next, is there a current process to dissociate solid tumor cells to allow the use of this technology?
RareCyte has not embarked on that type of evaluation, which would be, in a sense, I suppose, analogous to what's done by flow cytometry. Rather, there are some users that are doing some rather novel work in the tissue area to be able to preserve tissue architecture and still identify rare cells. So in this situation, one would use fluorescence staining of the tissue, and then using the same type of algorithmic approach to identify rare cells, identify those cells within the tissue, and then pick those cells. The caveat there is when cells are adjacent to other cells, it can be very challenging to get one cell. Rather, it's quite possible to get a few cells there. So it's not to say that this is not possible. I just would say that it has not been the focus of our efforts, but it would certainly be something that could be done. One could make smears from the disaggregated tumors and then try it this way.
Okay, thank you. What are the advantages of cell-based liquid biopsy over cell-free DNA analysis? Well,
that's actually a very timely question, too. As many of you know, the general term liquid biopsy is more tightly associated with cell-free DNA analysis currently than with cell-based analysis. And I think that a few years ago, I think the field generally thought that since the cell-free approach and evaluation of the plasma seemed so straightforward, that this would really be the future of genotyping. I think the advantages, I should just say this too, that the approaches, I believe, and I think many in the field believe, will be complementary. They are different tools, and depending on the situation, a different tool should be applied. Oftentimes, one hears this analogy of the difference between a fruit smoothie and a fruit salad. The idea being that cell-free DNA analysis gives the sum total of all the fruit that's been ground up in a fruit smoothie. One can identify mutations but not necessarily know what fruit they came from, whereas the fruit salad approach is being able to get individual cells, and now if there are different mutations identified, one can ask, are they all present in the same cell or are they in different cells? I think another potential advantage is that the DNA there is pure. There is no background DNA that requires bioinformatic analysis or any other method to separate. So I would say that there are going to be complementary situations in which one or the other will be, I think, potentially preferred, much like it is in the fetal cell analysis field right now.
Okay, good, thank you. Now, 4 plus 2 means that any additional 2 markers can be added, is that right?
So yes, what that means is additional markers can be added. Now, currently, there are, I would say, minimal constraints regarding species. So generally speaking, if your antibodies are standard rabbit or mouse species, then they should work quite nicely in the additional 2 channels. The assays, of course, require validation similar to any other type of immuno-based assay validation.
Okay, thanks. How small in size and what stage tumor can be identified with liquid biopsy in your practice, for example?
You know, that's another really good question. If you scan the literature, you'll find that as cancers, if you look at the whole spectrum of cancers from stage 1 to stage 4, epithelial cancers, you'll find the highest incidence of CTCs that have been identified in the stage 3 and the stage 4 categories. It's not that they don't exist in stage 1 or stage 2, but they are traditionally have been a lot harder to find. And I think the reason for not finding so many can be related to many different factors. One could be the overall sensitivity of the assay. Another one could be due to the actual volume requirements. If there is only one CTC that's present in, say, 20 mLs of blood, but the typical sampling is 7.5 mLs of blood, you can see it's a statistical question of how likely it is to be able to identify that cell. I think that one way that I think about the progression of cancer is that, you know, all stage 4 cancers were at one time the stage 1 cancers. All metastatic tumors were once CTCs. By this I mean distant metastases. So the sense that I have based on our own experience is that as the methods become increasingly sensitive, we're going to find more and more evidence further up the cancer progression scale or stage in which it will be very rational to apply these methods, perhaps screening in high-risk populations, and certainly the monitoring of recurrence after treatment. So even though my answer isn't definitive, and it does certainly depend on cancer type, I think that what we're going to find is that, in general, the later the stage, the higher the incidence of CTCs being identified, but increasingly we're going to find more and more situations where lower stage tumors have CTCs that couldn't be meaningfully evaluated.
Okay, thanks. When do you think that cell-based, another good question, when do you think that cell-based liquid biopsy will enter clinical pathology practice?
You know, that's another very interesting thing to think about. As a young biotechnology company, our approach is to develop the technology initially for research use, and then the next likely step is going to be to develop it for laboratory test use via CLIA laboratories, primarily in early adopting academic centers, and then eventually reach that point very likely via Companion Diagnostics, where we get regulatory approvals at the federal level. So I think there's probably a pretty good chance that within as early as the next year, there will be initial academic adoption in CLIA laboratories for the use of the RareCyte platform. And then I suspect that that would quickly lead to additional applications within the CLIA setting. I think once there's a formal approval of a test at the FDA level, then I think it will become much, much more rapid. The other possibility, too, is that if, in fact, we can apply these methods effectively to make non-diagnostic cytology samples into diagnostic ones, I can see the entrance of this technology into the standard pathology workflow very rapidly.
Okay, wonderful, thanks. Is there a current process to disassociate solid tumor cells to allow use of this technology?
Yeah, thanks, Rick. I think we just went over that question just a moment ago, but you know, I think you've given me just another idea to add one more bit of answer to that is when it comes to the actual tissue staining, there is a method that is currently being developed by one of our academic collaborators in which the tissue is stained with one round of immunofluorescence staining. This tissue is imaged, and then the stain is, in a sense, wiped clear from that slide. And then another round of staining is performed with a new set of markers, and then that is imaged. And this is repeated for several to many rounds. What that, in effect, does is generates really multidimensional, deep multidimensional information in which that tissue can be interrogated at a single cell level. And I think this is going to become a very, very interesting research tool to help understand the heterogeneity of both tumor cells as well as tumor microenvironment cells, including tumor-specific immune cells.
Excellent, and thank you. And thank you for saving me on repeating that same question twice. This is what happens when you break your routine just a little bit. Would it be possible to exfoliate cells from a fresh tumor and achieve a cytology in order to use this technique? If by exfoliating cells you mean something like making a touch prep or some other method in which you have free individual cells or clumps of cells in fluid, yes, that's quite possible. We have actually used versions of techniques such as thin prep as well as our own method to be able to take cells in suspension, spread them onto slides, and then evaluate them in this way. And continued efforts in this arena are actually ongoing. Okay, thank you. We do have just a few more questions, so if you have any questions out there, be sure and send them in using the chat box or the Q&A box. This is a great opportunity to get them answered. Next, can other cytology preparations besides fine needle aspirates be analyzed with this platform? Yeah, that's actually, it just follows on the previous question. We just touched on touch preps. We have done some initial proof of concept work, again with academics, looking at urine specimens and then processing urine specimens using the thin prep method onto slides, and then being able to stain with our standard 4-marker panel, identify the presence of spiked-in cells into human urine, and then be able to pick them and perform molecular analyses and demonstrate that, yes, in fact, we could identify and confirm mutations present there. So in a general sense, you can consider rare cell problems as being a few cells in the background of many, many cells, or another way to think about it is a few cells that are present in a very large volume. So both urine is
such a specimen type. Cerebrospinal fluid would be another, and certainly pleural fluids and ascites fluids could also be other types of fluids that would fit there. And I see, frankly, no reason why things like bronchial brushings and so on couldn't also fit within this paradigm.
Great, thank you. And following on that, can other rare cells besides cancer and fetal cells be identified with the RareCyte platform?
Right, I've mentioned a couple of times briefly the possibility of looking at rare immune cell phenotypes. I didn't show slides here, but once one has 6 marker channels available, one can start to subtype lymphocytes according to functional categories. And by using such markers such as CD3, CD4, CD8, and then other markers such as PD1, it's either TIM1 or TIM3, I'm blanking on it, but the exhaustion markers, one can find T cells of interest that are present at very, very low concentration within the blood. In addition, we've done some really fascinating work with other colleagues at the University of Washington identifying antigen-specific T cells, both CD4 and CD8, that react against the polyomavirus that is the causative agent of Merkel cell carcinoma of the skin. So I think this just reinforces the concept that if there is a rare cell that's present in a sample, and if there are markers that can differentiate that from other cells in the background, then we should be able to find it, whether or not it's a tumor cell.
Okay, excellent, thank you. Is it possible to stain for more than 6 markers?
Yeah, as we discussed just recently, this ability to be able to stain and then to unstain and then restain allows that possibility to stain for multiple markers and get the deeper level of information. At this point, we can only stain for 6 markers simultaneously, although I think our engineers are looking to see whether there are ways to be able to even extend that to additional channels.
Okay, thanks. Would you say that with the possibility of doing a touch prep on fresh tumors, this technique comes as an aid and even sort of a substitute to frozen sections?
You know, I'm thinking on the fly right now. I've never actually considered how this could be used as a substitute in real time for frozen sections. But, you know, actually, I think this might be challenging, and the reason for that is as the workflow is currently set up, the amount of time that it would take to stain on the automated stainer using our standard approach would probably be too long to be able to keep the patient in the operating room to be able to generate that result. I think it's an interesting idea, and I think what you'd have to do is come up with maybe a more minimal set of markers and maybe another approach that would allow the staining to be done very, very quickly. But right now, I think the major constraint would be the amount of time required in order to be able to get the surgeons the answer that they need. And currently, a typical run on the automated stainer will take, kind of like a regular IHC run, it'll take maybe anywhere from 1.5 to 2.5 or 3 hours. And so I think that would be an impossibility right there. But it's an interesting idea to think about, and I appreciate the question.
Okay, excellent. Thank you very much. And that is all the questions that we have, which is quite a number of questions. So thank you for generating all of those. Let's move on to close things out here for today. If you would like to write an educational article for pathology leaders, be sure and send it or just let Eric Heilman know. And here is his email address. Susanna has put that into the chat box. Thank you, Suze. And today's recording will be available here at Pathology Leaders/webinars. So Suzanne has put that into the chat box as well. So if you'd like to access the recording or suggest to your colleagues that they view today's session, this is where you can tell them to go. You can also view all sorts of other archived sessions from the Pathology Leaders webinar series at this website as well, and also register for upcoming events. If you'd like to receive PACE credit for today's session, when you close the event window, you will see the evaluation open in your browser, so be sure and leave your browser open. Don't close that when you close the WebEx window. Fill out the evaluation, send it right in. If we receive your evaluation within 24 hours, then you will be receiving your certificate of attendance as a link in an email within a couple of days after we receive your evaluation. If it takes more than 24 hours to receive your evaluation, though, it can take up to 2 weeks to send your certificate. So go ahead and send that in right away. If you are joining us as part of a group, whoever has logged in today will be receiving an email within about 10 minutes or so after the scheduled stop time of today's webinar. Be sure and forward that email to everyone who joined with you so that they can access their evaluations as well. Same applies to those of you who joined using a mobile device. You'll be looking for an email from messenger@webex.com. That email will have a link to the evaluation, so be sure and forward or save that email rather so that you can access the evaluation link, which is inside that email. Again, the email is from messenger@webex.com, so please keep an eye out for that. As I mentioned, your certificate of attendance will be sent as a link in an email. If you have any unanswered questions or you have any questions that come up following today's session, send them here. This is the same email address that Susanna has put into the chat box before. She's just put it in again, which you can copy and paste for access later on. We'd love to have you join us for the upcoming Pathology Leaders webinar on December 11th, Understanding the 2018 Reimbursement Changes in the U.S. and What They Mean to Your AP Laboratory. Very, very self-explanatory title. And our presenter will be Loretta Sales. So a big thanks to you, Dr. Kaldjian, for a great presentation today. Thank you.
Thank you very much. My pleasure.
And a big thanks to our friends at Leica Biosystems for the wonderful Pathology Leaders webinar series. We hope that you will join us again very soon for another Pathology Leaders webinar, that one coming up in December, for example, right here. And be sure and visit the Pathology Leaders website so that you can register for that webinar. So a big thanks to all of you for joining us. And if you celebrate Thanksgiving, we hope you have a very happy and safe Thanksgiving. And we will see you next time. Have a wonderful rest of your day. And thank you again, Dr. Kaldjian, for a wonderful presentation. Bye-bye, everyone.